Hello, everyone. This is Dr. Wiseman. Here's my contact information. This is our first lecture. It covers aspects of Chapter 1. And as always, if you have any questions, reach out to me via email. You can text me, or you would be glad to hear your questions during our synchronous sessions. We're going to try to limit these online, asynchronous lectures to somewhere in the 30 to 40 minute range. And I'm going to try to give you material to help assist you with the reading. It's because of the format of these classes, the ability to cover the material efficiently while you're reading is going to be critical. And so these lectures are meant to supplement that. But as always, if you have questions, I will make time for you, even if we have to make an appointment and speak in person, outside of the normal class time. So chapter 1 is really about the large themes. We actually use Campbell Biology for two different classes here at UND. For Biology 155, which is more ecology-minded, more holistic type biology, organismal biology. And then we use it for our Intro to Cell Biology. So this first chapter is really relatively unifying. It's about these themes that span biology, what make biology a cohesive subject, if you will. I know that many of you are early in your academic careers. If you are an upper level student, you probably already understand this. But even though there is a heavy factual component to this class, the key is that you need to be functional with it. You can memorize some of it, and yes, there will be some memorizing, but there's more than that. We're going to try to help you move in that direction. Sometimes it'll be straightforward, and then sometimes I'll have to work with you a little bit more. But they all encompass these themes, if you will, these general themes. One theme is that the phenomena that we're looking at is scalable. We can see some interaction at the atomic level, at the molecular level. We can see interactions at the organelle, cell, all the way through. So there are interactions at every level. There are emergent properties at every level, and there's energy flow at every level. So we're going to be kind of covering some of these themes here in Chapter 1, just to kind of get a start. So it's an integrative science. It's using chemistry. It's using psychology in some cases and physics. So there's many levels, and that's occurring at many levels. And so one of the barriers, I think, to folks enjoying biology is the fact that it can get complicated pretty quickly. That's because of what we call emergent properties. And that's saying that because we have multiple degrees of freedom, that multiple things are affecting a given situation, that means that there may be behaviors and outcomes that can't necessarily be predicted. So let's look at some examples of emergent properties. So again, we have emergent properties, and these things result from interactions, arrangements, and multiple inputs that change behavior. And it's not necessarily a biologic thing. We can have non-biological entities, and maybe that serves us well for the example that I'm going to give you. When we have the parts in the right places, we can have a functioning bicycle. We add a person on there, and that bicycle can certainly move in a given direction. But who would have predicted, based on this simple system, some of the things that people can actually do with bicycles? So as we add the ability, additional inputs, the complexity goes up quite dramatically, very, very quickly. Another fun example of this, and maybe some of you have these sitting around in your house, maybe your parents have something like this, would be a Newton's cradle. This is where you have several balls suspended and you can swing them back and forth. If you swing one, only one will go off the other side. If you do two, the two will go off the other side. Things that you wouldn't necessarily predict just looking at the simplicity of the system. Unfortunately, I can't stream other videos inside my presentations in this format. So I've included the link here, and this is worth your time. It's only a couple of minutes long. What they did was they took that Newton's cradle, but each of the different balls has a different length of string. So take a look at that. It's about a minute and 45 seconds. I've got this main video up here, or I have a second video down here. They're really, really cool. Just watch it for a couple of minutes and see what you might not expect in terms of the behavior when it comes to these just one variable here. All of these balls are identical in mass. And composition. It's just that the string is slightly longer or shorter in each of those different balls. Very fascinating. And that's an example of how biology can really throw people pretty quickly just by adding one or two variables. And just for fun, another perfect example of emergent properties, this time on mountain bikes, one of my favorite videos to watch. It's a lot of fun. It's only a couple minutes long. Worth your time if you enjoy videos like this. As I said, the concepts like, for instance, emergent properties and so forth are scalable from literally the smallest entities, atoms and subatomic particles, all the way up to biospheres, the entire planets, or perhaps the biosphere of our solar system, as if we happen to discover life on other planets in the solar system, or if we spread across planets ourselves. But it's scalable. And so what we're going to do is learn about all of these things by looking at the fundamental entity, which is that of the cell. And so these are the levels that we're going to be working with. Not tissues so much, that's more physiology, but we are going to be working primarily in these first three levels of organization in 165. The atoms and the molecules therein, functional groups, the organelles and some of the cellular, subcellular mechanisms, like for instance photosynthesis and respiration. So we're going to be looking at the molecular and the cellular level, as well as some of the physiologic needs of the cells, how the cell gets the job done. That's the goal here, because as it turns out, if you're interested in healthcare, this is where 99% of the action is. Yes, we have behaviors and population influences and environmental influences, and those are important, but they are feeding back into this level, and that's why this class is that's why we put this right up near, early on in the biology curriculum at the 165 level, because of its preeminent importance in all aspects of biology, the functioning of a cell. Again, we're going to try to move you in a direction. We're going to try to move you in a direction where we're working towards process. Certainly we can work on function, and that's kind of the teleologic approach. That's kind of the memorization side of the approach. And yes, there is a role for that. But what we're going to try to do is get you more into the how. So for instance, we're going to get into molecules and electrons and things early on in this unit in particular, than unit 1. But when we start to move into unit 2, like when we get into cell signaling, signal transduction, we're now moving into what we call the mechanistic approach, or the reductionist approach. We're gonna scaffold in this class. We're gonna give you the pieces of the puzzle, and then we're gonna start putting those pieces together into mechanisms and pathways. So we're gonna learn about those electrons, those functional groups. We're gonna learn about proteins, carbohydrates and nucleic acids, and then we're gonna show you how they get the job done, at least initially, that how, if you will. So we're transitioning from a purely memorization approach into a more functional approach, because understanding that how gives you goals for therapy. It gives you the avenues for how you're going to approach a specific problem. For instance, if you have too low blood pH, or if you have too high blood pH, the manifestations of both of those are similar. Okay, you have the similar. They have signs and symptoms in common, but they are exactly opposite in how you would treat it. You'd want to raise pH if they're acidotic, and you're going to want to lower pH if they're alkalotic. Your body's a little better able to handle acidosis than it is alkalosis. And so again, the alkalotic side might be a little more significant. That's why we have to learn the how on this, because it's not enough just to know something's wrong. We've got to figure out how to get it back to where it's right. Another scalable theme, like emergent properties, is this idea of energy transfer and the change in energy. Like, for instance, chemical energy to physical energy, kinetic energy to potential energy, and so forth. A lot of what we're doing inside cells, a lot of what we're doing in terms of organ function and so forth, is regulating and controlling the flow of energy. We take energy in order to turn it into something that's useful inside our cells, primarily ATP. Maybe other molecules and so forth that we use for structural purposes and storage purposes. But nevertheless, we're controlling energy is what we're doing. Energy in terms of keeping things going here and moving materials both in and out of the body. So that's a scalable concept. We see that at the subcellular and the molecular level. We see that at the biosphere level. One major aspect is what we call work, and that's basically kinetic energy. So moving, growing, reproducing, it's things in motion. That's work. But we're transferring energy. It's becoming chemical energy, turning into kinetic energy or heat. A lot of energy moves through the body and then is eventually released as heat. So it flows through the cell, it flows through the body, it flows through an organism and or an entire ecosystem. Let me give you some examples of what I'm talking about from the last slide. For instance, we need glucose and other types of simple sugars in order to create ATP, which is one of our energy producing molecules or energy storage molecules inside cells. Well, we expend energy to get that glucose into the body, and then we transport it around the bloodstream, and then we get it into the cell. And that all requires energy of some kind. But the last thing we want to do is, if we have glucose moving into the cell, is to have it move right back out. And if all we had was the diffusion doing that, where we had glucose diffusing in, it would diffuse right back out, or we would get even concentrations of glucose on both sides, and then it would stop. So we don't want that. We don't want to lose glucose, and we don't want it to reach equilibrium. So we're going to expend energy so that glucose only generally moves into a cell, and only a couple of different cells in the body actually have glucose that leaves the cells. Another example, and this is an even better one, I would guess, if I were to see each and every one of you, which I wish was possible, I'm sorry, that's not the case. Even if you only weigh 100 pounds, and if you weigh 250 pounds and you're a lineman on the UND football team, all of us burn about 150 pounds of ATP every day. Well, we recycle ATP, so that's why you don't weigh 150 pounds, alone in ATP. But nevertheless, that's an energetic process. We are converting energy from one form into a stored form in the chemical bond of ATP to the tune of 150 pounds of ATP per day. So energy transfer is really, really important. So we've had emergent properties, we've had energy transfer and transformation, and now our third theme is the idea of structure and function. And again, this is scalable. Okay, if you will do me a favor and just put your hand up in front of you for a second, why is that hand shaped the way that it is? It is shaped the way that it is so that it can interact with and manipulate any number of different structures in our environment, as opposed to, say, a cat's paw or a horse's hoof. Though, cat's paw has a different structure, and it has a slightly different function in the terms of what a cat uses its paws for, or a horse's hoof and how it uses its hooves, just like, an appendage just like our arm, in order to achieve its physiologic goals. The structures are different because the functions are different. Yes, there are similarities, and those similarities imply overlap in terms of function. Structure and function are almost inseparable, So very, very closely related. A leaf is thin and it's flat, maximizing the ability to capture light and to facilitate gas exchange in addition to that. We're always balancing the needs with the cons, the vulnerabilities, if you will. Structure of a bird's wing for flight. Not only does the bird's wing have to be appropriately shaped, but you have to have very, very light bones. So let's take a look at that. So here's our bird wing and a seagull here. You can't have very, very dense bones like, say, that we have in our bodies. If you look at a human body, we'd have to have some major structural differences in our body if we truly wanted to be able to achieve flight. First of all, our wings wouldn't be up at the level of our shoulder. We'd be having to fly like a, it wouldn't work. It would have to be closer to our hips where our center of gravity is. Secondly, our bones would have to be much, much less dense because that's extra weight. They sacrifice a little bit of strength. The bird's bones can be quite strong. Nevertheless, they are much, much more porous because of that need for less weight. And we see this in the aircraft that we build ourselves. We build aircraft out of the lightest and strongest materials possible because it's more difficult to counteract gravity when you have everything that's super heavy. And here's a really great example. This really takes it right to something that's near and dear to my heart, and that's pharmacology. And by the way, most of you, if you are going into occupational therapy or physical therapy or medicine or veterinary or any type of healthcare, you're going to be dealing with pharmacology at one level or another. Well, let's use a pharmacologic example. It turns out biochemically, our universe, our biosphere here on the planet Earth is left-handed. This is a drug that we're all familiar with. It's an opioid known as morphine. Very, very powerful, powerful drug that mimics endogenous proteins found inside our body. This structure right here happens to look a lot like what's called an opioid peptide. So structure and function. This thing alleviates pain, its function alleviates pain, and it's because of its structure. It interacts with receptors. On the other hand, we have the dextro. This is the right-handed, and we'll talk about left and right-handed later on in our biomolecules side of the equation. This is the same molecule, only it's right-handed, and it's that if you shine light on this molecule over here, it bends the light to the right. You shine the molecule, now the molecule over here, it bends it to the left. Hold your hand up and you can see that they're mirror images of each other. Okay, so this right-handed one over here is a molecule known as dextromethorphan, much less potent than our left-handed one and consequently much less addictive. It's a wonderful cough suppressant that's much less addictive, and so it's much safer to use than, say, morphine. Morphine will suppress a cough. People that have very, very severe coughs oftentimes will get a little tincture of morphine in there in order to ease the coughing reflex, but it's much more addictive and much more carefully regulated. Structure and function. Theme number 4 gets more into Bio 165 and so forth. But it may be self-evident. It's that the cell is the lowest level of organization that can perform all activities required for life. There are smaller levels of organization, certainly organelles. In fact, organelles in many cases resemble cells, but they can't perform all activities required for life. Viruses, like for instance our infamous COVID, it has to have a cell in order for it to replicate itself. It can't replicate on its own. So the cell is the lowest level of organization that can perform all the activities required for life, like growth and movement and reproduction and so forth, and communication in some cases when we're talking about tissues and cells working together and so forth. But cells are the lowest level. And all cells have one or two things in common, two things minimum. They all have a membrane and they all use DNA as their fundamental genetic information. We have DNA, we have RNA and other types of nucleic acids, but the DNA contains the heritable genetic information. As far as we know, we might learn tomorrow that somehow RNA is transmitted or something. That would be exciting. Nevertheless, as far as we know, DNA is the genetic information, and all cells have to have a membrane.
So let's look at an E. coli. This is an E. coli cell. Now this one you can't see a nucleus, even though we know it's a prokaryotic cell. It won't have a nucleus. It'll have DNA though that it uses to pass on information. And it has a cell wall, but it also has a cell membrane. Not all cells have cell walls, but all cells have a membrane and all use DNA as their genetic information. Got a nice enhanced electron microscope image here in scale. In scale. So on the right up here, this is a prokaryotic cell. This happens to be a bacterial cell. It's got DNA, but it doesn't have a membrane-bound nucleus. And so while we can see an enrichment of DNA in these purple areas, just like an enrichment here of DNA, these areas are called nucleoids. They do not contain a membrane. Over here on the left, in our eukaryotic cell, there's lots and lots of DNA here, but it's also enclosed in a membrane-like structure. The only membranes that these two cells have in common are around here and around here. And we'll talk more about prokaryotic versus eukaryotic cells, but that's at the very least. Prokaryotic cells and eukaryotic cells both have membranes and they both use DNA. And that's where then things begin to differ a little bit. Our final theme, theme 5, is a close relative of theme 4. If we said that we have to have DNA in the cells, well, what is the function of DNA? It is the way we transmit and store what we call heritable information. That is to say, an organism's characteristics are often inherited from that organism's parents, the genetic ancestors. And that's in the form of DNA, deoxyribonucleic acid, the double helix, if you will. And we're going to talk about this at length in the later parts of the semester. So we have deoxyribonucleic acid and we have ribonucleic acid. But DNA is the substance of the genes, and the genes are what we call a functional unit of the DNA. These are what confer Genes are what confer the characteristics. You can have DNA that's non-functional. In fact, a big chunk of our DNA in our cells is actually just repetitive DNA that has no known function or no identified function. It may have some function we just don't know yet, but we can identify genes because they have a common structure. We can identify active units of DNA in terms of what is going to be transcribed into usually proteins. And those proteins then allow us to, or allow cells to function properly. That is in terms of coordinating reproduction, growth, repair, metabolism, response to injury, and so forth. So a lot of functions there in a very, very tiny package. Just for a moment, just a little bit of trivia for you. Take your hands and hold them about a yard apart, maybe a little longer than a yard. That's a meter. Every cell in your body that has a nucleus has that much DNA inside of it. It's absolutely amazing. In fact, there are information scientists that are looking at using an artificial form of DNA for high capacity data storage. It's pretty amazing stuff. And again, when we get to the chapter on biomolecules, we will be talking about nucleic acids and DNA in particular. We're going to talk about DNA replication later on in the semester, and mitosis and meiosis, and gene expression. So we've got some ground to cover with DNA this semester. This is a really, really great story about a woman who had no intention of having such a major role in medical history. In fact, she probably didn't know, she was never aware of it while she was alive. But that said, she is still technically alive. She died of a very, very aggressive ovarian cancer back in the 1950s. And her cells, which we think of cells as biologic entities, and that's true, but cells serve a very, very important role in medicine because her cells became what are known as the HeLa cells, and we still grow HeLa cells today. Henrietta Lacks, for her name. She was never intended to be revealed in terms of her name. It was just a sort of a mistake to use the HeLa, but her cells were responsible for developing the polio vaccine. Her cells have traveled into space and have led to all sorts of wonderful, wonderful things. But there's also some very, very important ethical issues. A lot of the HIPAA laws and patient privacy and the release forms that you sign today are in part due to what happened to Henrietta and her cells. If you get a chance and you're really interested in medicine, you should read this book or listen to the audiobook. It's not a difficult book to read. There's also an HBO film about it. It's called The Immortal Life of Henrietta Lacks. It's a wonderful book, even if cell biology isn't your thing, because there's really, really good stuff about medicine and science and history in there. The latter part of chapter 1 involves talking about the scientific method. And unfortunately, the book does kind of takes a little bit of a turn there. That's okay. We're going to cover this a little bit in our labs and in some exercises that will replace what we used to do, which was a lab manuscript. So we're going to work on this idea of making observations and then forming and testing hypotheses. A lot of times people and students get alienated by this aspect of this class because we do this automatically. We will observe something and then make a guess about it and then go for it. What we're trying to do here is help organize that process so that when you talk about it and you're working with other people, and that's as important as anything in your careers, is being able to work with patients, work with other caregivers, people in your department or in your field. The idea of organizing your thoughts is critical because for whatever reason, our thoughts inside our heads make perfect sense, but until they're out there in the world, they may not make sense to others, and that's just the plain old truth. So this part of the class is about helping you organize those thoughts, helping you put them down in an organized and methodical way, so that you're easily understood and you're more effective in your communications. So we're going to be doing some labs this semester. We're still working on the schedule a little bit. We're waiting for the labs to come in, but we're going to be doing some inquiry labs. You can do those with your at-home lab kits. And we're going to reinforce this idea of scientific process. And this is the basic order. You start with an observation. You have an experience with something. You see it, you look at it, you observe it, and then you make a guess about something it's doing or there. And then you go through the process of testing and evaluating. So at its base level, it's observation, making a guess about it, and then seeing if that guess is actually correct. You also may have heard before coming to UIndy, and you might hear it every so often here at UIndy, is that we like to stress what we call critical thinking. Another way of saying critical thinking is analysis of data and creativity. What we see, what we observe, and then what we do about that observation. We think about it. We consider the alternatives and the plans, what's causing this phenomenon that we're observing. And we try to design and execute an experiment to test whether or not our analysis of that observation is incorrect. And so you're going to see different levels of complexity when it comes to the scientific method, but it's always these three basic things. So my task for you is to say, well, these are the basic things. Can you identify those? And then can you add on as we decorate this Christmas tree a little bit with additional steps? But it's all connected. It's all connected, and that's what makes biology so special, and physiology so special, and medicine so special, but yet tricky at the same time. We may be dealing with a problem with their heart, not realizing that something else, like for instance, their psychological state or the enzymes in their lungs. We're going to talk about COVID-19, and we're going to talk about the ACE enzyme. Well, the ACE enzyme is actually found in the lungs. Well, that could affect hypertension and blood pressure, which could affect the heart. So the goals for the lab part of the class, We're going to try to develop the ability to do a good hypothesis, one that can be tested. We're going to work with math. I know that math doesn't come easy. I know it didn't come easy for me. What we're going to try to do is work with a little bit of just basic analytical process using just a little bit of math, a little bit of statistics to get us by. I hope that at some point you will take a statistics class. I think it's a very, very useful class no matter what type of career you're interested in. It will help you to identify when people have done the analysis correctly and when they're trying to snow you over, if you know what I mean. Fake news, putting a spin on it, if you know what I mean. We're in a very terrible time that way. We're going to work on graphs a little bit, and so let's talk about some of those types of graphs we might be using. Okay. Lastly, we're going to try to reinforce those writing skills. Writing skills don't come easy. They come with practice. Learning to adapt comes with practice. Scientific writing is nothing more than just writing in a given formal style. And I'll give you some examples of these articles going forward, including this week. I have a couple of articles in mind. And what I'll do is I'll show you a very basic aspect of scientific writing that we're going to work towards. And the book's going to talk a little bit about types of data. There are two general categories of data. We have qualitative data that's more descriptive rather than measuring. Like for instance, our example here, every fall maple trees turn red before they die and fall off the tree. Well, how many leaves turn red and how many turn yellow and how many turn brown? Well, they say red, we're just saying qualitatively. We don't have any level of extent, if you will, no quantity, if you will, and that's where quantitative data may come in. What is the maximum intensity of the redness before a maple leaf falls and dies off, or dies and falls off? How many leaves turn red and how many don't? How many turn yellow or some other color? That's quantitative data, and that's where the real money is. Qualitative data is really, really nice early on. And it can be important, especially if you're describing something new, but don't let it be the substitute for the real meat and potatoes of science, which is quantitative data. Quantitative data allows for more objective analysis. Qualitative data can be very objective, it's possible, but it's harder to be objective in qualitative data. Here's an example of some exceptionally important qualitative data. This is Jane Goodall, still alive today, but she did some very, very groundbreaking work with chimpanzees and their behaviors and so forth. And she created a qualitative data set that is still used today in behavior theory and social dynamics. But it was qualitative data that she did. It wasn't really quantitative so much. So here we have some quantitative data. Quantitative data is most often graphed where you have an independent variable, that is to say something that maybe we manipulate, or something that changes in the experiment, that may perhaps be driving change in the experiment, or something that we control. Like for instance, let's say this x-axis is temperature. That is an independent variable, and that's really tricky. It's even hard for me to say sometimes. But if it's on the x-axis, odds are it is an independent variable, something that is manipulated or controlled, or something that is observed over, say, time or so forth. On the y-axis, we get what is known as the dependent variable. When we tell a story in a graph, and that's what we're doing, when we plot a graph, what we're trying to do is find a story in it. What we can see here is that for whatever reason, these dots in this XY scatter are lower than the ones here in that are depicted as X's. For whatever reason, the independent variable and the dependent variables are causing some sort of different outcome. Okay, but independent variables on the x-axis, dependent variables generally on the y. And again, it's the same story. Let's just take it into a bar graph this time. The dependent variable is the type of diet that these canaries are fed. So they have different types of diets, and that leads to different amounts of body or food intake. We can see that the canaries seem to like A diet better than either B or C. So each bar is showing a distinct variable, or a change from the original control experiment. independent variable, excuse me. Dependent variable here is food intake. This is known as a histogram. And a histogram shows you the number of events at a given dependent variable setting. So how many students got a 1 on their quiz? How many students got a 10 or so on their quiz? It shows you the population in each dependent or independent variable group. Okay? It quantifies a distribution of a variable over a range of values. Here's an example of a line graph. Again, independent variable on the x-axis, dependent variable on the y. And we can see that males have on average over time, and these probably are mice or something, is showing that the males had a higher body weight than the females. If you want to have some fun, this is a wonderful video of a very famous scientist. He was actually the one that came up with the likely reason for why the shuttle Challenger exploded in 1986. A guy named Richard Feynman, who won the Nobel Prize in Physics. This is a short video of a lecture of his. You can see how far we've come in terms of lecture, talking about the idea of forming and testing a hypothesis. And if you want to hear it in a different voice, this is worth a couple of minutes of your time. And if you did watch that video in the previous slide, you will see that he calls the hypothesis a guess. Well, what is a guess versus a reasonable guess? A guess can just be out in the blind, and that is a kind of hypothesis, but a true guess is not a hypothesis because it doesn't have observation. Okay? That's what makes a reasonable guess different and a hypothesis different from just a blank guess. And I want to encourage you to continue working on your observation. Don't wait for the answer. Look for the observation. See if you can't come up with a possible guess before either the answer or the conclusion arrives. That's the difference between a hypothesis and just shooting blindly. A good hypothesis can be tested. Not all hypotheses can be tested, but a good one can. And that's what we want to do. We want to be able to test it so that it can hold up to observation or experimentation. Are leaves green on the trees because of a divine presence? That is a reasonable guess. Leaves are amazing structures, you know, and if you have faith, that might be considered a reasonable guess. Unfortunately, it's not very testable. It is very difficult, or nobody has yet come up with a definitive yes/no test for the divine presence. That said, are leaves that shape because of the genes in their DNA in their cells? That is testable. Okay? That is testable. So that's where we're trying to go, is to help you work on your observation before you make a guess, and to develop tests that will allow you to see if your guess is correct or incorrect. Okay? So if it's correct, awesome. It confirms your hypothesis. Maybe you got ambiguous data out there. It's hard to say. Sometimes it is. So we might have to develop a couple of different tests to see if they corroborate. Or the worst is if we have something that isn't supported by the data. Then we might have to come up with an alternative hypothesis, something else. For instance, my leaf analogy, there's a divine presence that made that leaf, or there was DNA in the genes. The DNA in the genes would be an alternative hypothesis. If you can't falsify a hypothesis, it does not necessarily mean that it's true. Just because you can't falsify it does not necessarily mean, and you might see this going on with COVID, is that scientists are hesitant to go, we proved it. Anytime you hear the word prove, that usually rings alarm bells with scientists, because we like to keep it ongoing. We might understand something fairly well, but there may be more to learn. For example, if you have a flashlight bulb and you replace it because it wasn't working, that it supports the hypothesis wasn't burned out, but maybe it was just that the bulb wasn't inserted correctly, and when you put that other one in, you did. So there are possibilities here, both of which are testable. Take your old bulb, put it back in, make sure it's put in correctly, and test that hypothesis. And as much as I would love to say that science is clean, it's not. But it's also a beautiful not clean. It can be a very, very interesting not clean. In fact, most of the major discoveries over the last century have occurred because something didn't turn out right and somebody went, oh, what's going on there? So while there's this lovely idea that we could observe and hypothesize and do that, but it's always a little more messy than that. The idea is, when can we address that mess and when do we have to kind of work through that mess? I know that as students you like to have certainty, but now that we're moving into the university level of education, or you've been here for a few years and you're getting ready to go on to graduate school, the going into graduate school and into advanced studies always means that you're going to get more gray areas. There's going to be more in between. We're going to try to stay away from that as much as possible in this class, but it may be times when we get a little bit in a gray area. And just the key is to roll with it. Try to figure out why it's not working out the way that it is, and keep moving with that hypothesis-based science. So observations, hypotheses, results, and analysis, but it must be repeatable. Otherwise, it's very, very difficult to advance from there. It must be repeatable. It must be testable. But it's a cooperative effort, and not everyone in the room will necessarily agree. But the idea here is to keep the rigor in place. If something isn't necessarily correct, having various viewpoints in the room really does help give you stronger and better rigor. Most everybody nowadays works in teams. Oftentimes with graduate and undergraduate students. And so working in teams, we're not going to have that opportunity as much as I'd like. What I'm going to try to do this semester is assign you into discussion groups and working groups to help come up with responses to challenges and ideas so that you can kind of share your viewpoints and get that idea of working in cooperative approach. Not as strong as I'd like it to be, but that's just, that's the circumstances this semester. But good communication is important. It's good for working with others. If you guys are having trouble, if there's something I'm doing that's driving you nuts, I run a judgment-free zone. Let me know what's giving you trouble. I will do whatever I can in my power to help you be as successful as you can be. Okay. So we went just a little longer than I'd like. I was aiming for half an hour, but we're right at around 40 minutes. But just to summarize a little bit, what are the scalable themes we talked about today? I would ask that you go ahead and write those down and work on finding examples rather than just the examples that I gave you. Can you find your own? Can you find an example of an emergent property in your life? Can you see examples of these themes? If you guys want to see a cell and see a cell membrane, crack open an egg in your kitchen and look at that, and then look at that egg yolk. That egg yolk is one enormous cell. The largest single cell that we know of on this planet is the yolk of an ostrich egg. Okay, the largest egg and largest single cell. So let's find those themes out in your world. Let's make lemonade out of the lemon. You guys are stuck in your rooms. You guys are stuck at home. See if you can't take some of this and move it into your life. The more you do that, I guarantee you, the more it'll stick in your brain, and the more interesting it'll become. What scalable themes, when you are doing your other classes, can you see these themes? If you're working in volunteer work, can you see these themes? The goals of the scientific process, the key aspects, the difference between qualitative and quantitative data, the types and the graphs and the types of data presentation. Those are sort of the key things that we talked about in this chapter and the book describes. So take a look at that chapter, read through it, try to read, take good notes so that you don't have to read it more than once, because that's a very, very big time sink. I do not want to discourage you from reading. It's important. But the way to speed up is to slow down. Take notes so that you don't have to read more than once or go back to a specific section if you're having trouble. That's it for today. Thanks for your time. I will talk to you when we get to Chapter 2 if I don't see you in one of our synchronous sessions. Take care.